Hey, Hammy here. Today we're going to go over section 7.4, on which we look at the regulation of gene expressed. Okay, when and how genes are turned on or off and making proteins or not within our cells. Okay, it's important to remember that most of the cells in your body, have all 46 chromosomes. They have all the copies of your genes. And it's estimated that you have about 20,000 different genes on those 46 chromosomes. Now, does every single one of your cells need to be coding all 20,000 of those genes? No, think about it. You have tissues in your body that are very specific. Skin cells have a certain job that they do. Your stomach cells, liver cells, pancreatic cells, the cells in your eye and your retina, they do very specific things. So they only need certain genes to be active, the ones that they need to function correctly. Okay. If a gene is active, we say that gene is expressed. Okay. Gene expression is using that gene to make the end result, which is a protein. Okay. Gene regulation. Okay. Gene regulation is whether that gene is on or off. Okay. Controlling it to say when and where those proteins are needed in the cells. Okay. So remember the central dogma. We're going from DNA through transcription to RNA, which is then. Okay. So that's all stored on your DNA, but now the step becomes, when do we go on with this rest of the central dogma? When does the cell need certain things? Okay, most gene expression is controlled by regulatory proteins, that bind to regulatory elements near the promoter region of a gene. Okay, remember the promoter region. If you look down here at our DNA, the promoter region is where RNA polymerase is going to attach and then take off and transcribe that gene, okay, which makes an mRNA transcript, which will then go out to the cytoplasm and take those instructions and build the protein. Okay, so a lot of times nearby that promoter are regulatory elements, and you have the same clip or the same picture in your notes. Notice that there are repressors, just like the name sounds. Notice that these are negatives. Repressors are things that will slow down or stop expression of a gene, whereas activators will, notice they're pluses, activators will speed up or turn a gene on so that it makes even more proteins. Okay, so you've got sort of negative elements and positive elements. First, we want to look at prokaryotic regulation. Remember, prokaryotes do not have a nucleus, okay, but they still have their own genome. Okay, they have one big, you have a bacterial cell. Inside that cell, and they don't have a nucleus, but somewhere near the center, they'll have a big loop of DNA that's got all their genes on. Again, they don't need to make all their genes all the time either. Scientists have found most of their genes are controlled by what we call an operon. Okay, it's a region or area of the DNA that consists of several genes that make proteins for a specific function. A famous one, is called the Lac Operon. You can see the name right here, Lac Operon. Lac comes for these three enzymes down here that are used to break down lactose. Lactose is a milk sugar, which the bacteria can use, the E. coli can use for energy. But now think about it. If there's no lactose around, does it want to waste time and materials to make enzymes to break down something that's not there? No. So it has this mechanism by which there's a regulatory gene kind of upstream, that codes for mRNA transcript, that goes out, and they have kind of simplified ribosomes in a prokaryote, and it makes a protein called an active repressor. That repressor comes and sticks in the operator. So here comes RNA polymerase to read the genes to break down lactose, it's like a block or a stop sign. So if there's no lactose, this repressor puts a stop and says, nope, you're not going to code for proteins today. Okay, it blocks it. Now, if lactose is present, okay, look what happens over here when the operon is turned on. Lactose or allolactose binds to the repressor, so it fits in that spot right there, and it causes it to kind of bend or change shape, pulls it away from the operator. Now what can our RNA polymerase do? Whee, it's happy. It can now get past the roadblock, okay, and it's going to read those 3 genes, and now the bacterium can make enzymes to break down that lactose. Okay, if the lactose starts to run out, okay, the enzymes will start to come back and break apart these lactoses here. Once the lactose is all broken down, the repressors will go back to their original shape, go back to the operator, and shut off the RNA polymerase. Say, whoa, stop, we don't need any more enzymes. Okay, so this operon is a way that the E. coli can turn a gene on or off, and they have found other operons like this in prokaryotes and other bacteria. And it's just a way for them to turn genes on and off as they need a certain gene product. In eukaryotes, okay, that would be us, plants, fungi, okay, other animals. This is a much more complex than the simple operons. Usually they involve enhancer regions of DNA, distant regions of DNA that produce certain kinds of proteins. Those proteins, encircle this on your picture in your notes, we call transcription factors. Okay, you might see that term again. These transcription factors, okay, they code for mRNA, which make these transcription factors or proteins, okay, that often bind to a region that we call the TATA box, T-A-T-A, thymine, adenine, thymine, adenine. Okay, so there's a region with a bunch of T's and A's. There's a binding protein, okay, that sticks into this region. And notice it's right before the gene right here. And if we need a certain product, or we need a lot of a certain product quickly, or a gene needs to be turned on, okay, they code for these proteins that make a binding protein. And then all these transcription factors all bind together at that TATA box. Notice what it does to the DNA. It causes it to bend. Okay, that bend really helps RNA polymerase right here get in and start to transcribe the gene. And the more that this gene is transcribed, okay, the more protein product is going to be made. Okay, so it's more than just one repressor that binds to the promoter or the operator. Here we've got a whole host of transcription factors and binding proteins that kind of clump together, twist the DNA, and be able to have that gene be read more efficiently and quicker, because we need more of that protein product. Some interesting group of genes that scientists have found are called homeobox genes. Sometimes you may just see them referred to as Hawks genes, right? Like go Hiland Hawks. Sorry, bad joke. Okay, homeobox genes code for certain regions of the body, and they turn certain genes on and off during development. And this helps develop our body plan, and it puts our arms in the right place, our eyes, our nose, all our organs in the right place, and helps us develop properly. They've done a lot of research with fruit flies, and they've been able to do some like weird science fiction type of stuff. Okay, they identify where these Hawks genes are for the different body regions of the fly. Here you can see them in different colors here. And when they start messing with these, this is a picture over here of a fruit fly, in which you can see those don't look like normal antenna or mouth parts. Okay, those are actually legs. Okay, they got legs to grow kind of on the forehead of the fruit fly. Okay, and so with some really weird stuff that they can get to do. Now, obviously, just on fruit flies and other kinds of lab organisms, just to see what effect these different Hawks genes have. We may think evolutionarily, you know, that's how we went from like a dragonfly that has like 2 sets of wings Okay, dragonfly here, some eyes up here, to other insects that just have two wings. Something was mutated, or a mutation happened in those hawks or control genes early in development. It very much changed the phenotype. Kind of the last topic to talk about when we talk about gene regulation is this topic of cancer. Okay, cancer is kind of scary for a lot of us. We've been touched by people who are affected by cancer. Okay, cancer is our own cells kind of going haywire. And that's why it's tricky to treat. It's not like you're fighting off some outside bacteria or something that's infecting your cells. Okay, it's mutations that arise usually in one of two places: tumor suppressor genes or proto oncogenes. Okay, now tumor suppressor genes are kind of like the brakes to the cell cycle. Okay, so normally when you get a cut or something like that on your hand, okay, the cells will grow until they grow back together, and they usually have what we call contact inhibition. They'll grow until they start to touch, till they close the wound or the gap, and then that's the signal to them to put on the brakes on the cell cycle, stop going through mitosis, and to stop dividing. Okay. If you have a mutation in a tumor suppressor gene, one of the famous ones is called P53. Okay, it's like the brakes going out on your car. So this illustration down here, and without the brakes on your car, crash. Okay, they're going to grow out of control and form a tumor and take the place of normal functioning cells. The other one, Proto Oncogenes, they're like the gas pedal. So let's say, like we said, you need to heal an injury. The Proto Oncogenes stimulate the cell cycle. They cause those cells to start growing faster. A mutation here in the Proto Oncogenes, they then become what we call oncogenes, which are cancer genes. Okay, and they're like mashing down, that would be this one down here, where they're mashing on the accelerator and not letting off, like the accelerator gets stuck. And again, these cells will grow out of control and form a tumor or a mass, and they disrupt the normal functioning tissues at that place. Okay, so remember, cancer is when gene regulation kind of does not work. It's going without limits, and that makes it tricky. And scientists are, currently trying to find ways that we can treat those cells, find these genes, and knock them out, or help them not to get mutated or to turn into cancer-causing genes in the first place.